you all for being here. This is a really good turnout. I'm thrilled.
We're going to go ahead and get started, even though I think a couple of folks are still trickling in. I do want to actually start with a little bit of bookkeeping or housekeeping, since I know folks are interested. There's a sign-up sheet here. So if you're here for extra credit for a class, this will be out on the table when you go get dessert and coffee after the talk. And you should put your name on it, and then you check which class you are enrolled in and want extra credit for, and then I will take care of that for you. And then my Chan-Norris scholars, I'm checking you off as you come in, but will you please just check in with me before you leave this afternoon? So make sure that I got you if you're here, okay? Okay, so I want to welcome everybody to the first of two of these Chan-Norris Distinguished Lecturer Seminars. This is part of the Chan-Norris Scholars Program in Experiential Learning. And so for anyone who doesn't know me, I'm Dr. Sarah Swope. I'm in the biology department, and my area of expertise is ecology, evolution, and conservation. But I'm also the director of the Chan-Norris program, in the goal of which is to create these experiential learning opportunities for our students. So each year, scholars participate in a variety of events, and two of them are these invited seminars, and these things are open to everybody on campus. I do want to say that this program, the Chan-Norris program, which has brought Dr. Jani here from Hawaii, is generously funded by Annette Chan, who graduated from Mills College in 1965, and her husband, Evan Norris. So under my directorship, the program has this theme of global change ecology, and there are four major drivers of global change in the world right now: climate change, habitat loss, invasive species, and emerging infectious diseases. And then each year, we focus on one of those drivers, and this year it's emerging infectious diseases. But of course, all of these things interact with one another, and in February, our second seminar speaker is Nick Pilfold, who is going to come talk about how climate change is driving an increase of diseases in polar bears in the high Arctic. So if you're interested in that, I look for advertisement for that. But tonight we are thrilled to have Dr. Andrea Jani here with us from the University of Hawaii, and she's going to talk about her really important work on this emerging disease by a fungus that we call BD that's been decimating frogs the world over. So Dr. Jani earned her bachelor's degree from Cal right up the road, got her master's degree at Arizona State University, and then did her PhD at UC Santa Barbara in Sherry Briggs' lab, where she started this work on the chytrid crisis in frogs. She did her work up in the Sierra Nevada mountains, which are just beautiful, and you'll see some really nice photos of that in this talk. And that work was published in some of the highest tier journals out there. She's since moved on to the University of Hawaii, and I had to write this down, where she is a researcher at the Center for Microbial Oceanography. And she also teaches a disease ecology class every year there. So I invited her because I think her work is absolutely wonderful, but I also thought that it would appeal to every one of you, no matter what your interest in biology is. So she uses molecular and genomic tools to understand microbial communities, to understand how these microbial communities influence susceptibility to disease, and all of her work has a conservation application to it as well. So I just want to let you know, before I hand this over to her, I just want to let you know that there will be a Q&A question here after her talk. So if you have questions, please feel free to ask them. I'll be circulating with microphones. And then there will also be a reception with dessert and coffee afterwards, and please go chat with her. She's very friendly and will be happy to answer your questions. Okay, so with that, I want to hand it over to Dr. Andrea Jani.
So thank you so very much. I'm thrilled and honored to be here today. Super excited to kick off the first Chan-Norris Scholars lecture series. And thank you, Dr. Swope, for inviting me. Thank you, all of you, for being here on a Monday after the holiday weekend. And so I'll go ahead and start to talk to you about frogs and bacteria. So the title of my talk is Bodyguard or Bystander: Do symbiotic bacteria or microbes protect endangered frogs from a deadly disease? And so I'm going to try to take you through this journey that I went on to try to answer this question, which of course by the end you'll see is not yet answered. But we'll go through the part that at least we have so far. Does this work? Okay, good. So many of you may have heard of mass extinction events. These are events that have been documented in the fossil record, during which the rate of extinctions far exceeds the background level. And because the rate of extinctions rises so high during these events, we have biodiversity loss. So you can actually see in the fossil record these massive losses of species during these mass extinction events. And so there have been 5 documented in the fossil record. The most recent was the Cretaceous extinction, which is when we lost most of the dinosaurs. But it turns out that we were probably in the beginning of what might end up being another mass extinction. And you can find many sort of figures like this one that I'm showing. This is just one example. This is showing the bird species extinctions per century, starting with up to mid-1600 all the way through 2000. And basically, don't worry about the tiny numbers on the axis, but the point is that the number of species that go extinct per year or per century has increased tenfold over this time series. And one of the really alarming things here is that unlike these mass extinctions that we have observed in the fossil record, these extinctions are caused by one other species, which is us. And not all species or not all groups of species are equally impacted by these threats. And among animals, it turns out that amphibians, amphibians are especially hard hit. So 41% of amphibian species are threatened. And I'm going to talk about just one of these today, which is the Sierra Nevada or mountain yellow-legged frog. It's actually 2 species, but until a while ago they were considered one species. So they're very similar, and for the purpose of this talk, I'm just going to talk about them as one. Very cute little frog here. The males croak when you catch them and complain. They smell like garlic. Very fun to work with, but in serious decline. So this is a map showing the extent of their populations, which have declined over 95%. So they're only left in about 5% of the places where they once were. In this map, the red dots are the extinct populations, and the yellow dots are those that are remaining. And actually, this map is about 10 years old. Make a new one because they're now these yellow points. So the situation is pretty dire for this species. And one of the reasons, so Dr. Swope mentioned these threats to biodiversity, including climate change, invasive species, and that is one of the problems here. And this invasive species was introduced by humans. This is a trout. It used to be that above about 6,000 feet in the Sierra Nevada mountains, there were no fish in the lakes. And that's where these frogs exist. That's their habitat. And around the 1800s, or during the 1800s, gold prospectors and loggers or timber workers brought fish with them, some say even in their coffee pots on their mules, so that they could have something to eat. So they released these fish in the lakes in the Sierra Nevada mountains, and they were able to establish. After that, the fish continued to be stocked, actually by airplane by the California Department of Fish and Wildlife for recreational fishing. And so the lakes in these mountains have become very well stocked with fish, and it turns out that what fish like to eat is frogs. So these trout, they eat the tadpoles, they'll eat small adults, and if those adults that are big enough to escape predation, they have to compete with these fish for the rest of the food, which is insects. And so in lakes where fish are found, there are no frogs. And so they've driven the decline of the mountain yellow-legged frog in most of its habitat. So in the 1990s, a researcher named Roland Knapp and his colleagues tried an experiment, actually a conservation attempt, where they went in and laboriously removed these fish from some of these lakes. And it's always difficult to undo, always much more difficult to undo what we have done. And so it takes about at least 3 seasons of having a field crew go out there. They spend the whole season. So they hike out to this lake, they set up their camp, and every day they would set out these nets on the these places are inaccessible to people during that time. So when the researchers come back, when the snow and the ice melt at the end of the winter, in some places they came back to count the frogs and to monitor them again, and they found scenes like this, just dead frogs. And the culprit was a disease. So this is batrachochytrium dendrobatidis, which is a mouthful, but it's a chytrid fungus, which means it's an aquatic fungus from the chytrid group, and it infects frogs. It was only described around the year 2000, and is the only chytrid fungus known to infect vertebrates. So most chytrids are a very old lineage of fungi. Most of them are just aquatic fungi. Some of them will parasitize algae, or some of them just feed on pollen and organic matter. So this is really weird, because one of the questions is, why would one of these fungi evolve to attack vertebrates? We don't know, and that's an active area of research. But these chytrid fungi are aquatic fungi. They start out as a zoospore. It looks kind of like a sperm. It swims around in the water until it encounters a susceptible amphibian. And when it does, it invades the skin. It goes intracellular, so it goes inside the skin cells of the frog or the salamander, where it forms this sporangium. So these sort of pot-like structures here. These purple and pink pictures are histology slides where they've thin sectioned the tissue and stained it, and so you can see this is this frog skin. So these are frog skin cells, and then here are these chytrid structures that are why they're called chytrids in the little pot. So here's another. This is a scanning electron micrograph of the same structure. And so in the skin, they form these sporangia where they produce more zoospores, which are in here, which are then released from the skin back into the water, and then they can either turn right back around and infect the same frog or swim off to another one. And when frogs become heavily infected, they develop chytridiomycosis, which is the disease that's caused by the BD infection. By the way, I'm just going to say BD, not chytridiomycosis, so we can't say it too much. So when a frog becomes infected heavily, it makes the skin become really thickened and disorganized. And the skin is super important for frogs. They use it for respiration, they use it for electrolyte balance, and so when it's compromised, they start to lose electrolytes, and eventually they go into osmotic shock and they can die of that. So nasty, nasty disease. And one thing that's really scary about chytrid is that it does what we often think pathogens shouldn't. So pathogens shouldn't drive their host extinct. And the reason for that is that if a parasite depends on its host to survive and reproduce, and it kills its host, it's kind of shooting itself in the foot, right? So if they kill too many of their hosts, if they drive their host population extinct for many parasites or pathogens, that should spell the end for them. And so pathogens should evolve to not be too nasty. And yet, during this time when these chytrid outbreaks started to happen in the Sierra Nevada mountains, they documented these mass extinction events. So lakes that have been full, thousands of frogs previously, and then the researchers would come back, and sometimes over the course of a single season, all of the frogs would be gone. And so this here, this detail, depends on its host to survive. This probably is why chytrid is able to do this, because it doesn't only depend on this frog species. So the BD fungus can rely on what are called pathogen reservoirs. So even if it kills all of the frogs in a single lake, it can still hang out. And the places it hangs out are either literally in the water. So this is a physical reservoir where the pathogen can wait. And it can live for weeks, maybe more, in this water without encountering a single amphibian. So it can just hang out and wait. It's in no rush to find another live host. Or even worse, it can persist in other species of amphibians that can get infected but don't get sick, right? So that seems like a good thing, that you're not affected by this pathogen, but it's not good for your neighbors, because these frogs, this is a Sierra Nevada tree frog, and it can be infected. It can still be going around with its business, but it's still shedding zoospores. It's shedding chytrid fungus into the water, right? And so that's how it's possible for this chytrid fungus to actually drive the mountain-meal legged frog populations extinct. So this is a really scary situation. outbreak where a lot of these hosts die. But in some cases, not often, but in some of these lakes in the high Sierra Nevada, you see frogs that are infected with the same fungus, and they're doing okay. So these are called enzootic populations. And this is where the fungus is present. The pathogen is present. You can swab these frogs and test them for infection, and most of them, 80, 90% in some of these places, have this frog infecting have this fungus infecting their skin, but they're okay. They're not showing outward symptoms. You don't see dead frogs everywhere. And so, of course, the big question is why? Why is there this difference in outcome in some places? And more importantly, how? How are these frogs managing to survive and escape disease even though they're infected with the same pathogen? So the question is, how can some populations of frogs coexist with this nasty pathogen while others are driven to extirpation? And so there's a few working hypotheses. All of these have been and are active areas of research, and they're not mutually exclusive. The first is that differences between frogs might explain this pattern. So maybe frogs have different genetics in different areas of the Sierra Nevada, and some of them are more resistant to the disease. The other side of that coin is that it could be differences in the fungus itself, right? It's one species of fungus, but there's genetic variation among individuals in any species and any population. And so maybe in some areas we have a nastier, more virulent version of BD than in others. Could be differences in the environment. I mentioned earlier that BD is an aquatic fungus, right? So part of its lifetime, its life cycle, it's just hanging out in the water. It's hanging out in the lake. And there are zooplankton, things like Daphnia and rotifers, that can eat cucurbit fungi. So maybe in some places you have more of these predatory zooplankton. These are hypotheses that have yet to be proven or disproven. And then finally, those microbes. And of course, this is the part that's near and dear to my heart, and the part that I focus on. And the question is, do symbiotic microbes differ between persistent or enzootic and diotic or epizootic frog populations? So like humans, frogs are full of microbes. They have bacteria and fungi and viruses all over them and all inside them. And on their skin especially, it's moist, it's mucousy, it's a great place for bacteria and fungi to live. And so when a chytrid fungus needs to invade the skin, it's going to encounter this existing community of bacteria that are already there. And you can imagine that some communities are harder to invade than others, right? It's a community ecology question in some ways. And so it's possible that some populations of frogs have different bacteria on their skin, but those are somehow more defensive. Okay, so this is the question that I really wanted to answer. This was the first thing that I did. I set up an experiment or a study, a survey, and I planned to go out to 10 different populations in the Sierra Nevada. So here's California, here is Yosemite, here's Sequoia Kings Canyon National Park, and I'm showing here some of these populations of frogs. The green pins are frog populations that at the time of this study were persisting with the disease, right? So they're coexisting with BD. In red are populations that were experiencing crashes at the time, so they were experiencing active outbreaks and they were experiencing population declines. And in blue are populations that at the time were not yet infected, but the prediction was that they would probably also crash when BD arrived there. And so the plan was to go out to all 10 of these populations and sample the microbes on the skin of these frogs, and to do it multiple times, right? So because you can get seasonal variation in the microbiome of any animal, and so you have to get enough replicates so that you can sort of overcome that variation to see patterns that might relate to disease status. And so we would go out to these populations. I was going to swab a whole bunch of frogs in each of these places. And from the swabs, because the bacteria and the fungus both exist on the skin, in the skin, on the skin, and in the mucus, you can just swab them. So that's one of the nice things about it being a skin disease. And so you can get a look at the BD cells or the infection status of each frog. You can also see what their bacterial community is that they're carrying around on their skin. Can everyone hear me okay?
If you can.
And so here's where the fieldwork began. And this is some of the most fun I've had in my life, also some of the most difficult work I've done in my life. This is the Sierra Nevada mountains. This is just one of the lakes that we worked at. And a lot of these sites are far, far from even any trail, so you have to find them using a map and compass. And I got to spend whole summers just out in this environment. And so this is kind of what life was like during the field season. Lots and lots, oops, I'm so sorry, I keep pushing the wrong button. Lots and lots of hiking, big heavy backpacks, lugging these around. Some of these sites are 17 miles to get to, 3,000 miles elevation up, back down again, back up a few 1,000 miles in elevation. Sorry, not miles, feet, pardon me, goodness gracious. And so lots and lots of hiking, and then living out there in tents, anywhere from 1 to the longest trip I took was about 11 days. Carrying our food in these bear cans so that the bears aren't tempted to dig around in our packs. And then this is Marta, she was an undergraduate assistant working with me, and she's cooking her food here. Here's her little camp stove. So we carry everything with us. We traipse around in the Sierra Nevada all summer. It was great. Sometimes it wasn't as great, or some parts aren't as great. There's a ton of mosquitoes, and that's actually why you can see Marta's wearing a net on her face, because they drive you bonkers sometimes. And we try not to use DEET or any insecticide because we're working with these frogs, right? So that the mosquitoes are just coming for you all the time. Lots and lots of rainstorms, a lot of lightning storms as well, which is scary because sometimes you feel like you're the highest thing out there, right? So lightning strikes is coming for you. And then even hailstorms sometimes. A few times I woke up in the morning and there was snow around my tent. So not always the best conditions, but a lot of fun. And then the challenge is to bring the lab to the field, right? We need swabs for swabbing the frogs. We need these little scales for weighing how big they are, and these calipers for seeing how long they are. We need sterile tubes. We need lots of gloves. We carried so many pounds of gloves. And then once you get to your field site, so you hike out to your lake, you set up your little lab in the field, right? So you've got a notebook, we've got a big bag of gloves here, a swab that's drying, there's a field notebook. And then sometimes you have to get creative too, because sometimes you set up your little lab out here, and then you start to walk around the lake to catch your frogs, and by the time you catch one, you're 20 minutes away from your little lab. So we would also carry, this is a blow up of my head. You can see, I hope you can see, there's these little swabs, I stuck them in the band of my hat, and each one has a little tube with it, so once I swab a frog, I break off the swab in the little tube, and that way I don't always have to go back to my little home base. And so the first thing you do, you walk around the lake and you look for frogs. And when you see one, you catch it in your net, and then you take it out of the net with your gloved hand. And this is the part where they start to like complain and croak, and they grab the males, especially they have this little nub on their thumb. That's actually for holding up females. But they'll grab you with it and they'll kind of like push on your fingers, and it's not like garlic. And then you take your frog and you swab the underside, because that's where the fungus tends to concentrate. And once you swab them, you get their weight, which they don't tend to like this part. So this is a little field scale. And then you measure them with a caliper. We decide what life stage they are, adult, juvenile, and sex, and then they can go on their way. And they're usually pretty not too traumatized by this. And so then the swab is the goal for us, right? That's what we're really after, is the swab, because it has a mixed microbial sample. It has the bacteria from the skin of the frog, and it also has BD, if the frog is infected. So we can take this mixed microbial sample, we extract the DNA from it, and from that DNA, we can sequence the 16S gene, which is a marker gene in bacteria that allows you to distinguish species or frog species. Oh, sorry. Thank you. So the 16S marker gene allows you to distinguish groups of bacteria from each other. We loosely call them species. The technical term is taxonomic or operational taxonomic unit, or OTU, but basically they're more or less bacterial species based on their sequence. And then we can also use that same exact sample to look at the BD DNA and count by qPCR, by quantitative PCR, how many copies of the BD specific genes there are, and that way we know how heavily infected the frog is. And that's important because pathogen load, or how cells of the pathogen a host has can be really important for disease state. It's kind of like with COVID or the flu. The more virus particles you have, the sicker you're likely to get, and also the more infectious you are. So that's pathogen load. Okay, so the first thing that we saw is that in persistent or enzoitic sites, the frogs tend to keep low BD loads. So this is a graph showing one summer from July to September. The y-axis shows you the BD load. So this is a log scale, meaning that 2 means 100 BD cells were detected on an animal. And each of these lines is actually a population, so it's showing the mean BD load on frogs in each of these populations. The green lines show you populations that were persisting with BD, and what we notice is that the loads on these frogs never really go much higher than about 100 cells. So somehow they're keeping a really low load on their skin, and that's probably why they're not showing outward signs of disease. In contrast, here is a population that experienced a brand new outbreak during this summer that we were out there. And so this population at first had no BD, and then but really quickly, over the course of just a couple weeks, the loads in these frogs go up to 1,000 and even 10,000 cells. And this area that I've highlighted in pink, this is the lethal range. This is where frogs die. So again, BD load is super important. Disease outbreaks can move really fast, and in these resistant populations, the frogs keep low BD loads. And then here is the main punchline for me. This is showing you that resistant and susceptible frogs have different microbes on their skin. And the way we're showing that is an ordination plot. So I'm going to walk through this kind of slowly. So if you had just 2 bacterial species on your skin or on the skin of frogs, you could graph the abundance of those 2 species. One is the x-axis and one is the y-axis. But these frogs have 50 or 100 bacterial species, and so how do you show that on a 2-dimensional piece of paper or a screen? So what we do is an ordination, which just takes that multi-dimensional data and squashes it down into 2 dimensions. And so the main thing to know is that each of these points is the bacterial community on one frog, and the closer together 2 points are, the more similar those bacterial communities are. So for example, this individual, this frog, had a bacterial community, which I'm showing as a pie chart here. And these are actually classes, not species, because there's so many species, if I showed them in a pie chart, it would just be a blur. So this frog has a bacterial community that's characterized by these certain groups of bacteria. And if you look at another frog that's nearby on the plot, it has a similar pie chart. So they have similar bacterial communities. And then on this other side of the graph, these 2 frogs have communities that are super different from these, but similar to each other. So when 2 are close together, they're similar. And the punchline here is that these black dots are bacterial communities from frogs that are resistant to ED. And these open circles, these sort of just outlined dots, are from populations where the frogs were crashing. And so the point is that the bacterial communities are different in these 2 kinds of populations, which is really exciting. Okay, so microbiome composition is correlated with disease outcome. I'm going to take a little detour now and talk about correlations, because this, no matter how long you do science, I feel like this issue comes up again and again and again. What do you do with a correlation and how do you interpret it? And so I'm going to start with kind of a silly example, which is that you can imagine that the number of shark attacks and the rate of ice cream consumption might be correlated. Right? So here is a made-up graph showing in red shark attacks and in blue ice cream consumption per month. And they both peak in July. And we can look at this example and say, well, that's probably not because eating ice cream makes you more likely to get bit by a shark. It's probably because in the summer when it's hot out, people eat more ice cream and people go to the beach, which you have to go to the beach and go in the water to even have a chance of getting bit by a shark, right? So that's a kind of a silly example, and it's not always that easy. And so correlation is just a relationship between 2 variables. And so here's a correlation between A and B. And that relationship can be due to a few different types of sort of drivers. So one is just coincidence. This is a graph, I love this guy's work. So this is Tyler Vegan, can go to tylervegan.com, and he even has a book called Spurious Correlations. And so he has all these really ridiculous examples of things that are correlated for no reason at all, basically, or for reasons that are unrelated to each other. And so here we're showing the number of babies born named Sarah, just like Dr. Swope. And here in red is the hectares of Amazon forest remaining. And these are very tightly correlated. But I'm going to guess that babies named Sarah are not driving deforestation in the Amazon. So that's a coincidence or a spurious correlation. There can also be similar responses of both of your variables to a third variable that maybe you didn't measure. And so the shark and ice cream example is one of these cases, right? Where both the shark attacks and the ice cream are responding actually to another variable. So in this diagram, C is weather. And there can be causation. You can have situations, of course, where your correlation is because one variable is affecting the other. If A is driving B or B is driving A. And the trick is to figure out which of these situations you have, and if it is causal, who's driving who? Who's driving who? Okay, so back to this correlation that I showed you. We know that bacterial communities on these frogs differ. So there's a correlation between bacterial community and disease outcome. And maybe that's because the bacterial community on frogs here, these are the disease-resistant frogs. Maybe before BD ever showed up, the frogs in these populations had different bacteria than these. And maybe it so happens that bacteria here are better at keeping a fungal infection at bay. And so in that case, the idea is that the microbiome is driving this correlation. It's suppressing infection. It's preventing disease. But here, I've shown exactly the same graph. So the points on these two graphs are exactly the same. And what I've done here is color them based on how heavily infected a frog is. So the cool colors are uninfected or barely infected frogs, and the warmer they get and into red means those are seriously infected frogs. And so what you can see is that as BD load increases, as this color gets warmer, the microbial community is also shifting across this graph, right? And so it's possible that actually what we're seeing is that when BD infects a frog, it messes up their microbiome, right? Their skin is a mess. It's thickened, it's disorganized, the mucus changes. And so maybe this difference in the microbiome is just a response to a sick frog. And so this is really important to tease apart because it affects whether and how we try to use the microbiome to try to prevent disease, right? If the microbiome isn't actually protective here, then we would be wasting our time to try to manipulate it to protect frogs. So in order to tease this apart, we did an experiment. We asked, can we change the microbiomes on susceptible frogs to make them resistant? So we went out back into this era and we collected water now from 6 different lakes. 3 of these lakes were inhabited by frogs that were crashing, and 3 of the lakes were inhabited by frogs that were doing fine even though BD is present. So persistent and die-off frogs. We took the water back to the lab and we put it in tanks, and we put frogs in these tanks. And then we asked how this treatment of water from different sources affects their microbiome. So can we shift their microbiome so that it is maybe defensive, is sort of the objective here. And then we infect half of the frogs with BD, which is not fun at all. But we were hoping that this could maybe help us find a solution for some of these dying frogs in the wild. And so we wanted to know, can we prevent them from getting sick, even though they've been exposed to BD, by having sort of shifted their microbiome to be more like resistant frogs? And I need to thank the people who helped with this, because this involved, it's simple in concept, but we actually had to haul almost 100 pounds of water back from each of these 6 lakes, and some of them are something like 8-hour hike to get to. And so these people helped me haul all this water back, and I could not have done it without their help. And so we found that indeed, when we take these frogs and we put them in tanks with water from these sites, we could shift their microbiome. So the aquatic environment and whatever is in there naturally actually shifted the microbiome of these frogs. And so here I'm showing another ordination plot. The green dots are the microbial communities of frogs that were sitting or living in water from persistent sites, and the open gray circles are the microbial communities from frogs that were living in water from die-off sites. So they shifted their microbiomes, not as cleanly there's still a shift and it's significant. And there's also, so this graph is now showing the richness of bacterial species, or OTUs. And so the frogs that were housed in water from persistent sites have higher diversity of bacteria on their skin. So that was super exciting until we saw this, which is that when we then exposed these frogs to BD, that difference in their microbiomes didn't actually protect them. So this is showing the log of the BD load over time after we exposed these frogs to BD. And the two lines are the frogs that were living in persistent water and the frogs that were living in die-off water. There's no difference here. So that was a huge disappointment. And furthermore, what we did see really dramatically is that BD infection shifts the microbiome. So when BD infects a frog, it changes the bacteria that are dominant on that frog's skin. So I squeezed this figure in here, and I want you to not worry about the numbers on the y-axis, but just to know that each of these tiny little plots is showing you a comparison of the abundance, or the relative abundance, of a genus of bacteria on these two groups of frogs in the lab. So the frogs that were not exposed to BD and the frogs that were exposed to BD. And remember, we're trying to ask if there's a causal link here. And what we see is that there is. So when frogs become infected with BD, the relative abundance of certain groups of bacteria increase and others decrease. So this is a causal relationship showing that BD infection increases the abundance of these two types of bacteria, Rubrovidex and Undibacterium, and decreases the relative abundance of all of these bacteria, Rhodococcus, Enterobacterus, Dendrotrophomonas, Pseudomonas, etc. And one thing that's really concerning here is that some of these groups, such as Dendrotrophomonas and Pseudomonas, contain species that were hoped to be defensive. So other studies have found that certain strains of Pseudomonas, for example, and Dendrotrophomonas, can prevent the growth of BD in culture. So not necessarily in frogs, that has to still be tested, but if you take an agar plate and you coat it with certain species of Pseudomonas, and then try to grow BD on it, it won't grow. And so what's really surprising to me is that actually in this case, BD is preventing or is driving down the abundance of some of these groups that we would hope would be defensive. I should say that genus is an extremely broad category for bacteria, right? So you can have differences in virulence. For example, Salmonella is a human pathogen, and even subspecies of Salmonella can be different in their virulence. So this is very broad and very crude, but it is still interesting to note. The other important thing here is that we saw that the patterns that we observed in the field, and that's what I'm showing here. So again, each of these little tiny plots is relevant to the same bacterial group, but now I'm showing field data. So I'm going back to that field study that I talked about earlier. And now BD load is on the x-axis, so as you go from left to right, these frogs are more heavily infected with BD. And on the y-axis is the relative abundance of bacteria in these same groups. And so in almost all of these cases, if the bacterium was affected by BD in the lab, then in the field it showed a pattern that suggested the same effect, right? We can't show cause in the field, but we can show, so here, Pseudomonas. Pseudomonas declines in response to BD infection in the lab, and in the field, if you catch a bunch of frogs and test their microbiomes, the more infected those frogs are, the less Pseudomonas they have on their skin. So that suggests pretty strongly that the same disturbance phenomena is happening in the field. And the reason that I'm making such a big deal of this is that animals in the lab and animals in the field are very different, and their microbiomes especially tend to differ. As soon as you bring an animal into captivity, its microbiome changes. And so it's really important to actually verify that what we see in the lab has any relevance to wild animals. Okay, so unfortunately, so far we see no clear effect of the microbiome protecting frogs, and we do see that BD can really mess with the microbiome. And so one more question that I had is, what is the resilience of the microbiome? So resilience is when something can come back to its original state, right? If you stretch a rubber band, can it snap back to where it was before you stretched it? How far can you stretch it before it breaks? And so I wanted to know, since BD messes with the microbiome of the these frogs, if you cure them, if they're able to recover from disease, will their microbiome recover as well? And so I had a chance to test this when collaborators at the San Francisco Zoo and UC Santa Barbara and the National Park Service and National Forest Service, they all worked together to do this other conservation action, which was unrelated to the microbiome. They were actually trying to vaccinate frogs. So they brought tadpoles into the lab from the wild, and then when they became subadults or adults, when they metamorphosed, they would expose them to BD and then cure them, with the hope being that because their immune systems have seen BD, when they release them into the wild, they'll be able to handle it better. So it's kind of like vaccination, except that it's not an injection, we're just sort of pouring some BD culture into the water with them. And so what I was able to do was, while they were doing this, sample the microbiome and ask, if you cure them of their BD infection, does their microbiome bounce back? So here's what we saw. This is a graph basically showing the sequence of the experiment. So time is on the x-axis, and on the y-axis is BD load in red. And in the purple line, we're showing the shift in the microbiome. So if the purple line is at 0, then there's no shift, and the higher it goes, the more the microbiome is changing. So at time point 0, they expose these frogs to BD, and you can see that pretty quickly the BD loads go up, and so do, and then the microbiome shifts in response to that pretty quickly, right? So the microbiome is changing here. And then they cure the frogs of their BD infection, actually the drug that's used for athlete's foot. And as a result, their infections are cleared, right? So this red line goes back down to 0, but you can see the purple line does not. So even though these frogs have recovered from their BD infection, their microbiome doesn't come back to where it was before, right? So the microbiome is still disturbed. And so what this suggests is that at least in these frogs, this species of frog, the microbiome either has weak resilience or it's slow. Like maybe if we'd given them more time, they could have recovered. And there's always so many what ifs, right? So, but at least in this experiment, it looked like the microbiome is not terribly resilient to this disturbance. Okay, so coming back to our original question, bodyguard or bystander? I think probably a little bit of both. We find that in wild populations, disease-resistant and susceptible frogs have different microbiota, and that the aquatic environment definitely affects that microbiome. But most importantly, we saw that BD infection itself really messes with the microbiome. So it really drives these dramatic shifts in these frogs' microbiomes. And we haven't even got into what does this mean for the frogs other than disease, right? Because the microbiome is not just there to handle disease. It has a slew of functions, which for these frogs, we don't even know what those functions are. But presumably, those are being compromised as well. And we weren't able to protect the frogs by shifting the microbiome, at least not in the way we did it, right? Again, there's always other ways you could try this, and I think that's one of the sort of frustrations, but also one of the hopeful aspects of research, is there's always something else that you can try. And so I think there's a lot still to learn before we give up on this, and there's still a lot of active research being done in all of these areas. So I want to close with a couple slides about how this is not just about frogs. So my work was very much motivated by conservation biology, right? By the motivation of wanting to come up with a solution to this chytrid outbreak and to these declining frogs. These are endangered frogs, and they're really important. But this is not just important for frogs, it's actually directly important for us and for our well-being as humans. And one reason is that frogs in these ecosystems are important for ecosystem services. And what I mean by that is we talked about how these frogs exist in these high alpine lakes. And amphibians, especially those that breed in water, are unique in that they move resources from the water to the land. So these lakes are basically bowls, they're basins that are in a landscape of rock. And so everything flows down from the rock into the water. And the frogs are one of the forces that bring those things back out. Because as tadpoles, they're taking energy from the lake, organic matter, nutrients, and then when they metamorphose, they take that out with them to land. So they're moving resources back up out onto the land, which is really important for this ecosystem to cycle properly. They're also important for predators. There are garter snakes, there are predatory birds, and coyotes that rely on these frogs for food. And a functioning ecosystem in the Sierra Nevada actually directly impacts us because we, in California, many of our big cities get their municipal water from the Sierra Nevada. Right, so this glass of water on your table, so Alameda County, Oakland, Berkeley, San Francisco, Los Angeles, San Diego, all of these places get water from the Sierra Nevada. And so we are directly affected by the health of these ecosystems. And then there's the question of how these concepts relate to our own health as humans. I'm sure that many of you have read articles or heard other talks that have talked about how important the microbiome is to human health. Right? That's become incredibly obvious and incredibly well documented over the last couple of decades. So the human microbiome is critical for training our immune system. It trains our immune system not to overreact and therefore helps us to avoid autoimmune and allergic diseases. Our microbiome is important for metabolism of food, right, and has been shown to play a role when you have certain types of microbiota. It can enhance the chance of obesity. And this can be transmitted. In mouse studies, they can take the feces of obese mice, feed it to lean mice, and those mice will become obese. So it really is the microbes that are driving that. And then also, we've even, others, not me, I have nothing to do with this, but people have shown that that psychological state, anxiety, can be affected by your gut microbiome. So understanding how the microbiome interacts with its host and with pathogens is super important for our health as well. And we face disturbances in our microbiome too. Just like the frogs face this fungus that comes in and wreaks havoc on the microbiome, when we take antibiotics, it wipes out certain species in our microbiome. It totally changes the balance, and it can make us susceptible to other diseases, to other infections. One very well-known one is Clostridium difficile. This is a bacterium that is present in a certain a proportion of the population, but in some people overgrows, and it often invades after someone takes a course of antibiotics. Right? So once your microbiome isn't in its normal state, it's easier for some of these pathogens to move in. And in the case of Clostridium difficile, actually, there's some really cool, I'll get to this in a second, but there are attempts at harnessing our own microbiome to improve our health. Probiotics is one, but I'm going to express my opinion, at least, that probiotics, at least in our current state, are way more marketing than they are evidence-based. Right? So they're also not regulated, so most of the time you don't know what you're getting if you buy probiotics. And there's very little research so far to show that commercial probiotics do anything at all for you, other than cost you money. But there have been proven therapies that have come out of microbiome research with humans. I don't know how many of you recognize this. This is a microbial fecal transplant. And it's basically when you take the microbes, the fecal microbiome, so the poop, from a healthy person, and you administer that to someone who is seriously infected with Clostridium difficile. So this bacterium, once it establishes, which often happens after a course of antibiotics, after a disturbance to your microbiome, can cause really, really nasty disease. People get severe chronic watery diarrhea, they get cramps, they can get severe inflammation of the colon. It can disrupt your quality of life, or even in some cases be lethal. And they're very resistant to antibiotics. So some of them, there are no more antibiotics that you can throw at them that will kill them. But a microbial fecal transplant will. So this is actually an FDA-approved therapy for treating antibiotic-resistant Clostridium infections. So that was a little bit longer than I meant it to be, but that's just to say that these questions about disturbance in the microbiome and pathogens interacting with the microbiome matter for our own health. So again, thank you so much for having me today. It's just wonderful to be here. I'm so excited to get to look at it, see all of you. I want to thank my collaborators. I want to thank Mills College and the Chan-Norris Scholarship Program, and Dr. Swope for inviting me. My funders, the National Science Foundation and the National Institutes of Health. A lot of the beautiful pictures you saw were actually not taken by me, but by Leonie Waldridge and Georgia glad to. And again, thank you for being here, and I would love to take your questions.
How come it isn't feasible to cure the frogs if we already have one?
Great question. So it is feasible to cure them. The problem is that, and it's actually been tried to go and catch a whole bunch of frogs and treat them with the same antifungal that they used in that study that I mentioned. The problem seems to be once you release them again, they just get reinfected because this chytrid fungus is just in the water.
Thank you.
What are future steps being taken in this field?
A lot. How much time do you have? So people are still, I should step back and say that this is one species, and chytrid was actually first discovered in Australia and in Central America, and there are species there that have gone extinct or are threatened. And so there are a lot of different sort of methods that people are looking into. There is amphibian arc in Central America, where under really dire conditions, they take the last of these amphibians and take them into captivity and try to figure out how to, maybe bolster their microbiome or immunize them so that they can release them safely. There's a lot of research being done on the microbiome in other species as well. And a lot on the genetics of the frogs. And actually some of the active research right now in the Sierra Nevada is looking at, taking frogs from areas where they're doing well and moving them to areas where the frogs have gone extinct. Right? And so the idea there is that maybe it's actually a combination of the microbiome and the frog genetics that is determining their ability to resist. So it's kind of like groping in the dark. It just continues to be. But people are looking into all of those hypotheses that I listed: the frog genetics, the fungal genetics, the environment. Temperature is a big one. There's definitely concern that climate change is going to increase certain infectious diseases. With chytrid, it may actually could help. We don't know. There was a study a long time ago that claimed that climate change was exacerbating BD, but that's largely been sort of refuted or at least challenged. But the fungus here has a very low thermal tolerance limit. It can't survive above about 27 degrees Celsius. And so in some places, frogs, people have even tried sort of helping the frogs get a behavioral fever and warm them up. And then one very drastic measure that's been taken is actually to drain an entire area and try to dry it out. But of course that has other impacts as well.
Kind of going further off of that question, would you think it's moving towards looking for cures or a vaccine, or are you just trying to build up resistance in frogs overall?
Yeah, I think you're kind of hitting on how disease management is different for animals than it is for people. So disease management tends to be much more on a population level, whereas with people, we're very concerned with every single individual, right? And we treat each individual differently. With the frogs, because we don't have the resources or really the cooperation from the frogs themselves to do it, a lot of times we have to look at ecosystem level interventions. So there was a push to actually try to augment the bacteria in the ecosystem. So let me step back a little bit. There's a particular bacterium, actually in the Sierra Nevada, called Chan-Norris. It's purple. It produces a purple pigment that kills BD. For some reason, even though it's there, it's not really doing this job of preventing BD infection in the field. And some researchers have tried bringing cultures of this bacterium to the field, catching frogs and dipping them, and then releasing them, with the idea that unlike curing BD with an antifungal, it hopefully will establish and stick around on their skin. That hasn't been terribly successful, but coming back to this idea of more ecosystem-based approaches, some people are talking about, maybe, what if we release it in the water? I think that probably the reason it's not abundant is because that is the sort of equilibrium for the ecosystem. So I'm not sure that's going to work either. And so unfortunately, right now, we really don't have any answers, any good answers.
So what got you into this research, and why did you choose frogs out of like all the keystone species?
Why did I choose the frogs that are what?
Out of like all the keystone species.
So I joined this lab from a microbiology lab. So here's where like the messiness comes in, right? So I didn't plan for this to be my PhD. I was actually in a lab that was a classic molecular microbiology lab. I was studying human pathogens and trying to understand where their virulence genes come from and whether predators in the soil might be like causing the evolution of virulence genes. And then for various reasons, my advisor left, and I switched labs into Sherry Briggs' lab, and they were working on these frogs, and I thought, oh, microbes on frogs, no one's doing this in the system, and it seems like it could be important. So that's honestly why I ended up doing this. It was just sort of like the intersection of like circumstance and luck, and I feel so lucky because I absolutely love this work, and I'm so grateful that it ended up that way. But it wasn't like any like smart plan on my part, it just kind of happened.
I had an advisor in graduate school who called that fortuitous stumbling. Fortuitously stumbling into something really wonderful. Okay, given the time, I think this is a good place to end. So let's thank Dr. Jani one more time. Can all of my Chan-Norris scholars check in with me so I make sure that I've got you checked in as being here? And there is dessert and coffee, and Dr. Jani is more than happy to answer any questions. So thank you all very much.